Welcome to the first module of the training in organic breeding courses in the frame of the LiveSeeding project. I am Adrian Rodríguez-Burruezo. I work as full professor of genetics and plant breeding from the UPB, and I also research partner of the LiveSeeding project. And today we will start with this first module of these courses. Well, as you have seen in the survey, in the invitation for the registration, this training in organic breeding courses is made of 5 modules aimed at different issues in breeding, starting from the easiest or the basic ones, which is the one of today, plant genetic resources management, levels of plant genetic resources, etc. Okay, this will be the one of today, but we are also in the frame of other 4 modules. Module 2, which will be presented next Monday, February 3rd, aimed at phenomics, approaches and tools used for genetic resources and breeding materials. Module 3, which will be aimed at breeding methods fundamentals. Module 4, development and application of molecular methods in organic breeding. And finally, Module 5, organic heterogeneous materials design and development. The last one will be presented at the beginning of March. Okay, so the one of today is Module 1, aimed at plant genetic resources, is also made in 3 units. The one I will present right now, which is Unit 1, aimed at public institutional seed banks, plant genetic resources collection, conservation and exchange, okay, during probably half an hour, okay, we will make a break 5 minutes before, 5 minutes later. At 11, then will be the turn at 11:30 of Unit 2, which will be presented by my colleague Vladimir Meglic from Slovenia, who is also present now, aimed at pre-breeding from genetic resources characterization to their use in breeding programs. Then when Vladimir finishes at about 1 p.m., we'll make a lunch break of 1 hour and a half to restart with Unit 3, presented by Costas and Ricardo Bocci, aimed at community seed banks. The seed banks which are managed by farmers, organic, small, medium farmers, collections, exchange culture of seeds and land races. Okay, and it will take between 2:30 to 4 in the afternoon. Okay, so we will start first with my unit, public institutional seed banks, plant genetic resources collection, conservation, and exchange. So welcome to all of you. Welcome to the first unit of the first module. I will present most of the presentation, and I will have also the support of my colleague Eva Solbes, who is the co-curator of our UPB seed bank, which is internationally recognized, and working with the others from Wageningen, from AU, DRC, from Taiwan, USDA, etc. We are collaborating for decades. And well, firstly, which we have planned for today within this unit, will be during my presentation and combine it with my colleague Eva, as is planned as a dynamic mixture of a main body of presentation about the main topics of seed banks, seed management, exchanges, parameters, main knowledge, additional materials to complement the training of today. So you will have additional info at the end of the presentation. We will also use a guided virtual activity which will be managed by my colleague Eva. The unit has also a fast quiz, which will take for you. Okay, it's an activity for you. During 5-10 minutes, you will be asked to reply, to respond a fast quiz of about 4 questions related to the issues presented today. We'll have also a 5-10 minutes debate revision of the concepts show. Okay, and a final session of grow up, summary of everything, and proposed homework. Okay, I put emphasis in point 3 and point 5, which is the fast quiz, and also the proposed homework, which will be important for you, all the students, all the attending people. If you want to get a certificate or attendance, you need to provide your reply to the fast quiz and also the homework during next week. Okay, the fast quiz must be answered during 10 minutes. Okay, I will say, okay, now it's time for the quiz. These are the questions. Now you have 10 minutes to fill and give me back by email, directly by email. So you can write directly to these emails addresses, the mine, doctor.upb.s, and also the mail of Petra, my colleague from ETS Croatia, who is supporting the logistic of the presentations of today. So please, before everything, take note of these both emails, because you must answer the fast quiz directly to these emails, and in the email body provide the answer to the question, because it's faster. And then we will make a double checking of the people who has answered the fast quiz. Okay, so it's important right now that you take note of these both emails. Okay, and also the homework, which will be explained at the end of the presentation, should be sent also to these emails. But for the homework, you will have the whole week to prepare. Okay, but you need to send to both emails just for double checking. Okay, the fast quiz of today and also the homework for the rest of the week. So I will wait a few seconds for you to know the emails, and we will start the presentation. This will be also used in the next presentation from Vladimir in Unit 2 and Costas, Unit 3. I mean, send to their mails and Petra's email the answer to the quiz and the homework. Okay, they will remind you, but I am warning you also. Okay, so I hope you have write the both emails. We can start. So first, the first concept, so about public institutional seed banks. What's a seed bank? Okay, we must differentiate between, maybe some of you are familiar, you have had some subject aimed at seed banks, etc. But for all of you to start with a comfort point, what is a seed bank? We will start with the most basic one. We must differentiate between in situ versus ex situ conservation of diversity, agrobiodiversity in the case of cultivated plants. Well, while in situ mainly focuses on protecting organisms, plants, animal, microorganisms in their original habitat, ex situ conservation works on protecting this agrobiodiversity into internal protected habitats. As examples, in situ, national parks, protected areas or regions, the wilderness, but also on farm through the silent, the quiet work of farmers, generation after generation, season by season, growing their varieties, selecting the best fruits to take the seeds and restart the next season, and in this way preserving their varieties. This is on-farm conservation, okay, and is connected also with the unit of costas, unit 3. Okay, is connected to community seed banks, directly connected with farmers. This way of conservation, which is a dynamic conservation. Well, in the by contrast, we have ex situ, where botanical gardens, zoos are included, and also seed banks, which is the topic, the issue of today. Maybe many of you are not aware, but the botanical gardens and the zoos were born centuries ago, not only with the aim of showing something exotic, something beautiful from other countries, other regions, but also with a scientific aim of conservation, of exhibition, and for adaptation of foreign germplasm, foreign species to new areas. So these were the main aims of the botanical gardens and zoos at their beginning. Okay, but within this aim, seed banks are also part of ex situ conservation, and probably the most dynamic ones. Well, continuing with the concept of seed bank, seed banks, you must know that seed banks are essential to preserve agrodiversity or agrobiodiversity, which is called technically plant genetic resources, or the acronym PGR, for both the cultivated forms of species, the crops, but also their wild relatives, because they are also useful to introgress genes of interest when these genes are not, or these traits are lost or cannot be found among the cultivated forms. Okay. It's the materials are available to provide materials to farmers, sources of variation to breeders, or to perform genomic studies, etc. And a wide range, a plethora of applications. They are essential, the seed banks, to guarantee that agrobiodiversity does not lose. So it's very, very useful as a tool to mitigate the genetic erosion that can be produced in the real crops, but also they are essential to face future breeding challenges, like now the climate change. Okay, nevertheless, the limitation of the seed banks is that in comparison to on-farm conservation, the materials preserved in seed banks are preserved and multiplied every 5 or 10 years under ex situ conditions. So these materials do not evolve dynamically in their original habitat. As a result, we can find cases of genetic drift. Okay, I mean, the on-farm materials evolve dynamically year after year with the work of the farmers under the conditions they are supposed growing. Okay, so the impact of the climate change or other soil or environment evolution are affecting in the breeding process, in the evolution of the population. But in the case of seed banks can be then considered as some kind of security copy of these ancient varieties, okay, but do not dynamically evolve in the way of the on-farm conditions. So this limitation is important to have in mind. Okay. Then the part of collecting plant genetic resources. Well, in regarding collecting materials, there are several important terms. Any PGR, regardless their origin, their botanical status, cultivated form or crops, wild relatives, ancient races, breeding lines, etc., is named within a seed bank in the seed bank terminology with the term of accession. So any seed source preserved in a seed bank is called accession. And this accession is given a name or a code of the seed bank. Okay? These accessions or materials preserved in a seed bank with their code of the corresponding seed bank can come from exchanges with farmers or other seed banks, collecting expeditions to areas of natural occurrence or cultivation. Expeditions which can be performed at local, regional, national, or international level. But in such case of exchanging with other seed banks or expeditions at international level, you must take into account sanitary issues with care, because there are legal regulation in terms of quarantine and sanitary certificates in international or interstatal exchanges. My colleague Eva will provide some extra info about that in her slides. And in the international level, you must also consider that many decades ago, collecting in biodiversity hotspots, I mean the centers of origin of a specific species among foreign countries, was possible. Okay, there are very famous collecting expeditions in the 70s, even the 80s in South America, performed by people from the USDA, like Charles Rick, who explored all the tomato cultivated and wild relatives from the south of South America to Mexico. Okay, nowadays, since the signature of PGR protocols, international protocols, it has become difficult or impossible. Okay, so this must be also taken also with care. Okay, before passing to the next slide, I must also tell you, I forget at the early beginning, that if you have any question, okay, to be more efficient, please put your question during the presentation in the chat. Then my colleague Petra will compile the questions. I'll make a summary for me to answer during the part of the debate or the questions at the end of the presentation. Okay, so please, if you have any question during the presentation, please put your question in the chat. Okay, we'll be happy to answer the questions today if we have time, and for those questions we have no time today, we will share the answer and the reply by email in the next days. So remember, any question you have, please put in the chat. Well, in case of collecting plant genetic resources, in terms of local materials, this is quite easy. In particular, if you are working within the same state or your nation or the region. For instance, in our seed bank, since the 70s, we have been performing collecting expeditions along the region of Valencia, from the north to the south and in the inland part. For instance, on-farm collection of Valenciano and other tomato landrace around the region and surrounding areas. This is to be organized in a more efficient way. Usually is pre-organized with local farmers to prepare radial trips from the headquarters to radial collecting trips in one day or maybe can be organized if we are going from the north to the south of the region in round trips, which can take about 1 week. In case of interstatal or, let's say, more complicated way, international prospection expeditions, okay, as I told you, many years ago, this was easy, but with the new international regulation and signatures among countries, international treaties, this has become almost impossible, okay? But what will happen years ago? Tomato, for instance, prospection expeditions were performed to, for instance, tomato wild relatives or capsicum peppers, which are original to South America and Central America, before the international protocols in Ecuador, in Peru, in Chile, Galapagos Islands, etc. You can see, as I told you, the very famous trips, as I told you, of Charles Rick or Charles Heiser, exploring Solanum diversity in South America and Central America. You have pictures here of people, of colleagues of me in the 80s, performing prospection of Solanum chesmani in the sea cliffs of Galapagos Islands, peppers in the center of Bolivia or Peru, tomato wild relatives in Peru as well. As I told you, many years ago, many decades ago, this was allowed. Now it's not allowed. Maybe it can be allowed if you go in a trip in an agreement with a university, a public institution within the country of origin, where the diversity is held, and also signing with the ministry, the corresponding Ministry of Agriculture or domestic issues of the country, you can perform a paid expedition for prospection to help the local authorities or the local researchers to find their hot spots of diversity of the species grown there. Okay, but obviously you cannot take the seeds out of the country. Okay. Well, and what happens once the seed banks, the seed sources have been collected, okay, or are received from another seed bank, or are received from a farmer which collaborates with the seed bank? Well, first, the materials, when it enters to the seed bank, obviously we perform, at least in the big seed banks, we perform a test against the main diseases, and also we perform seed germination trials. But before that, the material receives an accession code and a passport data. Okay, I will explain later which is a passport data. If coming from other seed bank, I mean the seed sources or new accessions come through a seed banks exchange from other seed bank we collaborate, the received by seed bank records both the new code, I mean the code corresponding to this receptor seed bank, but also paired with the code from the ancient bank. Okay, this is very important because it allows a efficient control of the flow among banks, but also avoid in the future replicates, duplicates, or redundancies, talking about the same accession, the same genetic source. Because many years ago, when this protocol was not established, many seed banks, maybe they were sharing the same accession, the same genetic source, but they were given different codes. So the researchers, maybe they can ask the code of the seed bank from Wageningen, this accession, and another accession from the seed bank of Spain. Okay, from the UPB, yes. But maybe they were talking about the same accession. but with different codes. To avoid these cases of duplicates for researchers, for farmers in the future, for seed companies in the future, this is very important to note the new code of the seed receptor seed bank, but also the code of the ancient. And they are paid forever and ever. They are called the coordinates. Okay. Then the materials are checked for the main diseases. They are stabilized and preserved in the facilities, as we will explain. Well, also as part of the conservation and multiplication in the seed banks, once the accessions are integrated in the seed bank, they are managed with 3 different types. This is very important, right, to remember. 3 kinds of data for each accession or level or information relative to each accession. We have the passport data, which are linked between different banks, as I told you. If this seed sources, this, accessions from the CGM from Wageningen, the USDA system, etc., or the AURDC from Taiwan, okay, this is the seed bank code, and this is the new one. These are the passport data, okay? I will provide more details in the next slide. Then we have also the characterization data, which are not are less usual, less available than passport data. They are mainly based on phenomic traits. Okay, I will provide also more info in the next slides. And finally, the evaluation data. These are the less common level of data relative to each accession. So I repeat, we have passport data, which are the first one when the seed sources become a new accessions in the seed bank. And in case they come from other seed banks, they pair the new code with the ancient code from the donor seed bank, the characterization data based on phenomic traits, and the evaluation data. And now we will explain a bit. Well, the passport data. These are the most common when visiting the webpage of seed banks. So as I told you, you will have a practical exercise today with my colleague Eva, surveying, okay, flying in the repository of an example of seed bank online. Okay, so the first data you will have visiting this repository of accessions of Solanum lycopersicum, I mean tomato, or capsicum peppers, or cucumbers, is the link to the passport data. Okay, so this is the first data you will have visiting a repository of any seed bank. Okay, this passport data usually include or should include in the most serious and important seed banks the origin, data relative to the origin, soil and agroecological conditions, okay, the location, and in the last years, the GPS coordinates, if available, the location or locality, the country or state where they were collected, the species, the presumably species, okay, the varietal type, because within one species you have different varietal types or of tomatoes or peppers. So this is also when collecting with the local farmers, for instance, is important to note. The local name, if it has local name, this seed source, and if possible, even pictures taken at the moment of collecting or provided by the farmer or the seed provider at that moment. Okay. Well, in the presentation, you have an example in these links how the accessions are ordered in our seed bank, the UPB, seed bank repository with passport data. This is an example. Also, I'm providing you in the presentation another link to the CGN from Wageningen, the repository of Fababin with the link for passport data. Okay. As I told you, you will actively visit with the survey of an explanation of Eva and example. Okay, in the next slides. So I'm putting emphasis in this in the different levels of info of the accession because it's very important. Probably is one of the core questions of this of the presentation of today. Okay, here is examples on how advancing and looking at the other repository, you find the info. Okay, here, this is the info you will find in our seedbank. If you tape okay, this accession, I'm interested in this accession. I put a link. Okay, we have these examples. The availability, the number of accession, coding of the national inventory, because we are linked to the national seed bank program. Okay, the number of collection, the genus, the species, Solanum lycopersicum, etc. So these are the passport data of one accession within our seed bank. Okay. And this is passport data. We have also characterization data, which is the second data, the second level of data. This characterization data are based on descriptive phenomic or phenotypic traits. The most commonly used internationally are listed and have been prepared during decades by Biodiversity International, which was previously called the IGRI, the International Plant Genetic Resources Institute, which now is called Biodiversity International. This is the link for Biodiversity International, and this link has several catalogs corresponding to a plethora of cultivated species and their wild relatives. For instance, this is the picture corresponding to the descriptors for capsicum. These descriptors is a catalog of listed phenotypic traits highly inherited related to plant growth and development, reproductive organs, seeds, traits, and characteristics. They are specific descriptors of each species and wild relatives. Okay, this catalog, these descriptors corresponding to each species are available online free and in several languages, not only English, because for instance, descriptors for capsicum peppers are in English, but you can also find, for instance, in Spanish. And many of these catalogs are in Spanish, English, Portuguese, or even Italian or French. So it is important also if you want to collaborate in a participatory way with your small or medium farmers or breeders in order to characterize the materials you are working with together, because you can work in the local language. These traits are usually recorded, the characterization data are usually recorded during the usual process of a seed bank when the accessions are multiplied for first time to increase the seed stock. Consider that when a seed bank collects seeds from collecting expeditions at national level or receive seeds from another seed bank, the amount of seeds can be very few, 10, 20, 25, 30 seeds. This is very few for a seed bank. So maybe in the next year, this seed bank receives the seeds, Then the seeds are multiplied to increase the seed stock, to see if the accession is more or less homogeneous, and this process is also take advantage to characterize thanks to these descriptors. So it is made both the seed multiplication and increase of seed stock, but also the characterization of these new seed sources, these new accessions. Okay, with the characterization data, the seed banks or the researcher linked to the seed banks, like my case, which who I work with peppers mainly, we use this multiplication or my own research activities. Okay, maybe I can ask my colleague Eva, I need 100 accessions of peppers from this country, and this country, because next year we will work in characterizing for agronomic traits, but also we can also take advantage for descriptors. Okay, so within our usual working process, we characterize the materials with this dozens of descriptors, and with this data and characterization, we can perform an international publication like this one, as an example of phenomic characterization of Casicum collections from the Spanish Center of Diversity, the heirloom peppers from Spain. On the other side, this info of characterization, which is not usual, is more usual to find them in international publications, but nevertheless, in some species, some crops, or some seed banks, this characterization data can be also found together the passport data, like this example of the CGN of Wageningen. Okay, the Saint Fababin collection of Wageningen has the species of the crops and their accessions ordered by codes and with the passport data, but if you explore, you can also find characterization data, which have been also loaded. So this is not quite usual, but some international seed banks, if they have time or they have fundings for that, can also add the characterization data to the passport data, but it's less useful. Okay, and finally, the third level of seed bank accessions data, which are the evaluation data. These are the less available. Why? Because these characterizations, in these cases, the accessions, the collections of accessions are evaluated through evaluations mainly performed by breeders and pathologists, because in such cases, the collection accessions are evaluated to very specific and agronomically interesting traits. I mean, results of screening for resistance. For instance, some of my colleagues can go to the seed bank of the UPB, or other foreign researchers can ask my colleague Eva about, okay, you don't mind, Eva, can you please send 20 accessions of Spain? And also you have other 20 accessions from South America collected in the 70s, or coming from the USDA, from these areas, because we want to screen against this pathogen, or to find sources of interest against this abiotic stress, temperature, soil salinity, water salinity, etc., or even quality traits. I mean, levels of acidity and sugars for taste, or for flavor, or vitamin or antioxidant levels. These traits, as you may understand, are more specific, are very very difficult to take. They are not usual or common characterization data or descriptors, morphological description. They are more specific and more difficult and difficult to take. Okay, so they are specific resistances, quality traits, or other agronomic traits of interest. These are the evaluation data. As they are taken in more difficult ways, they are included in breeding programs, in national or international projects aimed at screening in seed banks or in exchanging materials, etc. In this way, these data are usually published in journals or congresses or meetings. Okay. For this reason, they are not quite common in the repositories of the seed banks. Maybe you can find, but if you find them, this is because this seed bank has included this kind of trait within an European project funded for that. Okay, you are funded, the UPB, to screen, not only to characterize these collections for descriptors, common descriptors, okay, characterization data, but also for these pathogens. You are paid for that. Okay, but with the compromise, with the commitment of uploading the results. In such cases, you can also find the evaluation data, but these are the less common data present in the repositories. Okay. For instance, you have also an example of my publication in capsicum collections. Okay, evaluated under organic conditions. Okay. Organic cultivation of heirloom peppers, variation in the level of bioactive compounds and effect of ripening, published in +1 6 years ago. Okay, this is the most common for evaluation data, the level of antioxidants and yield in a collection of peppers in organic versus conventional practices, but also sometimes, as I told you, but this is not very common. Again, the example of the CGN from Wageningen, you have some characterization and evaluation traits for some capsicum peppers in the CGN seed bank. Okay, this is an example where it's very rare, but you can find a list of accessions, okay, checked against the tobacco mosaic virus, the stigma length. Okay, you can find that these accessions of capsicum peppers have been evaluated. I mean, now we have ordered in the CGN how many accessions have been characterized for this range of traits, instead being ordered the accessions one by one. Now we have the contrary. We have the order of traits and the number of accessions characterized
Adrian,
for
sorry.
this trait. Yes?
Someone asked in the comments, what does TMV and TSW mean? If you can just explain it right now. TMV, what does that mean?
TMV, tobacco mosaic virus.
And the next one?
Tomato spotted wilt virus.
Thank you.
Okay, this is an example of, okay, Wageningen. In the Wageningen repository, you can order not only the accessions, the list of accession one by one, and then we will explore accession by accession, which data we will have available. But on the contrary, you can also order by traits. So you can explore, okay, I'm just interested in the anther color, which is a descriptor, is a characterization, common characterization data. Okay, so we have 1,387 observations in 52 experiments. Okay, you can see here the number of observations or accessions evaluated or characterized for this morphological trait, this characterization data. But also in the list, evaluation data are included. For instance, the last two examples here tomato mosaic virus and tomato spotted wilt virus, which we have about 500 and almost 1,000 observations or evaluations. That means 500 accessions evaluated against tomato virus, mosaic virus, or tomato spotted wilt virus, 1,000. So you have both the list of accessions of one specific accession, or maybe you are mostly interested on, okay, I will explore in the CGN the accessions of peppers evaluated for tomato mosaic virus because I'm a pathologist. Then do you list within capsicum peppers repository by traits, and then you explore if there are data, there are observations for tomato mosaic virus screens. Okay, and then you type the link and you enter and you check which accessions have been evaluated for this. This is a case of evaluation data, which is very rare but possible. Okay, you can see that in this list they are mostly morphological traits, I mean characterization data, and a few of evaluation data. For instance, these examples of tomato mosaic virus and tomato spotted wilt virus. Okay, well, now another important issue about conservation and multiplication. This is very important. We must differentiate between the three blocks of species: orthodox species, recalcitrant species, and vegetatively reproducing species. Why? Because it will affect to the way they are preserved. For instance, the most common in the seed banks, the orthodox. The orthodox species are species whose seeds support the essential dehydration previous to conservation at low temperatures, because in the seed banks, the seeds are preserved at low temperatures. There are chambers at 3, 4, 5 degrees, but there are also chambers at under zero for longer preservation periods. And for that, you need to dehydrate as much as possible the seeds. On the contrary, the seeds can die because of ice crystals. And the species that support an efficient dehydration for this refrigerated or frozen conservation are called orthodox, and their seeds are preserved at low temperatures. And these are the most common in Wageningen, CGN, or our seed bank here at the UPB. By contrast, you have also other species which are called recalcitrant. Okay, I repeat, this differentiation is very important. Recalcitrant, their seeds do not support the hydration because the seeds die. If they are dehydrated, they die, and therefore cannot be conserved at low temperatures. Okay, they cannot support the hydration because they die. Okay, I don't dehydrate. Well, but the seed will die by chilling injuries. So they cannot be preserved under low temperature. That's it. To compare conceptually both kind of species, orthodox species, as examples, you have tomato, peppers, moss cucumbers, Phaseolus, brassica species that can be preserved in pots, as you will see, and under low temperature. Okay, recalcitrant species, those species which have, or most those species will have big seeds and fleshy seeds. For instance, avocados, for instance, mangoes, and other species with fleshy and big size, and tropical species. Okay, just you to have ordered conceptually both kind of a block of species. Okay, and finally the species vegetatively reproduced. Okay, there are species that yes, the seeds can be preserved dehydrated and refrigerated or frozen. Okay, but the problem is that to preserve the variety, they must be reproduced clonally because their genetic background is highly heterogeneous. So you need to propagate them vegetatively. Okay. Then it also affects to their conservation conditions. On the right side, you can see that orthodox species are the ones whose seeds are previously dehydrated and then preserved at low temperatures. While recalcitrant or vegetatively reproduced species are preserved through clonal methods, through arboretums, through botanical gardens, or using in vitro techniques. You can see here in the next slide how orthodox species are preserved and dried. On the right, you have examples on how to extract the seeds, the seeds of tomato or the seeds of eggplant. Every species has their, let's say, specific or particular extraction method, okay? Because in the case of tomato, it's better to extract directly, stay them for a few days in the juice, okay? And they extract with a mesh and water the seeds, while, for instance, eggplant are mostly extracted through cutting the fruit when the seeds are ripened, and then using a blender and filtering the juice. So each species has their own extraction method. Well, once they are extracted, they are, I'm meaning orthodox species, because it is the sample of today. Then they are dried at least 1 week at room condition. This is the most usual way. Then they are preserved, refrigerated, or as I told you, even in frozen chambers. Why? Because this is also important. The relation between temperature, relative humidity, and the period of viability of seeds. It is called the Harrington's rule among the seed bank technicians. This Harrington's rule says that seed longevity decreases 50% each 1% relative humidity increase, or every 6 Celsius degrees temperature increases, or the contrary. The lower the temperature of preservation and the lower of relative humidity, you gain a longer period of longevity. Okay, this is the reason for which they are preserved, refrigerated, or even better, frozen, because frozen provides a longer period of viability. Well, the seeds can be preserved, as I told you, refrigerated, but then you need to multiplicate in shorter periods. I mean, the higher the refrigeration temperature, you need to ensure the seeds keep a good seed longevity and vigor to be multiplied every 5 or 10 years. It depends on the species. Okay. By contrast, if you have facilities for that, preserve the seeds below 0, the period of multiplication can be larger, because the seeds at this temperature can stay their viability for longer periods. The lower the temperature and the relative humidity, I repeat, the larger the seeds keep their longevity and their vigor. Okay? And obviously, after each multiplication within the seed banks, it's very usual to test the seed viability of this new stock. Okay? All this info and this correlation between this core issue of the seed banks the relationship between relative humidity and temperature with seed longevity can be also found with more details in the example I put in this slide on the footnote. Okay, many publications, but the most the best explained one is the publication of the UC Davis. Okay, this is the link. And in the case of recalcitrant species, as I told you, they can be preserved vegetatively in many ways: arboretums, botanical gardens, gerplums, orchards, in pots, as you can see here, our collection or ornamental cacti at the seed bank, but also using in vitro techniques. Okay, I'm also giving you in the slide examples of the Kew gardens. Okay, the Kew gardens are not only very beautiful gardens in Kew, close to London. Okay, but also they are aimed at many research or seed preserving lines. They have their seed collection preserved in pots at low temperature, the seeds, but also live in collection. You have here the link, and also in vitro collections. Okay, and the same in the botanical gardens of Madrid in Spain. But I'm giving you the example of Kew gardens. Good. And now is time for my colleague Eva, who will explain you the issues relative to seed exchange, seed entry, seed exchange with other seed banks, with farmers or receiving for farmers, with seed companies, etc. The issue relative to material transfer agreements, okay, and sanitary issues. I'm leaving you, Eva, for this info, and also at the end of these 2-3 slides, she will provide you the virtual trip to one of the examples or repositories. Okay, so I give the floor. Yes, I give the floor to my colleague Eva from my computer, and I will be back once Eva finishes.
Okay. Hello.
Hello, we can hear
Hello,
you.
hello. Sorry, because we have had a problem with the connection with my laptop. So I'll try to explain with the computer of Adrian. So thank you, Adrian. And now I'm going to explain a bit more about how we work in our gene bank. Well, first of all, I'm Eva Solbes, and I work in the COMAB the COMAB UPB gene bank. And I'm going to explain a bit more about how we exchange or deliver the seeds. I would like to know that we are a publishing bank, that we have to relation with other actors, and we exchange seeds with farmers, farmer associations, seed company, another seed bank, and research centers, and researchers in our center and other researchers in all over Europe and international deliveries. But I would like you to know that now it's very difficult the exchange and the delivery of yes. I'm going to do with Adrian. It's very difficult. Yes. Okay. Okay. Okay, yes. Sorry, because Adrian is telling me the time. So I would like to know that it's very difficult now the exchange, the delivery of seeds, especially if you want to access to plant genetic resources out of a seed bank. The most important thing you have to know that our gene bank provides the plant genetic resources under the signature of the SMTA, that is the Standard Material Transfer Agreement, provided by the International Treaty for Plant Genetics of for food and agriculture that I'm going to explain in the next slide. And also our gene bank, also complies the European requirements for phytosanitary requirements. Another thing I would like to explain is that our gene bank, but I don't know if the other gene banks applies a fees for the delivery of seeds for seed company and research units, but never we apply these fees for farmers or farmers association, because we consider that is a way to return the work of these farmers to for us. They many times they give us a lot of varieties, a lot of crops or species. So we never apply these fees for farmers or farmer association, or in case that another research center collaborate with us, we have to talk about the fee, the fees also. So the next, you can put the next. The next slide is about the standard material transfer agreement. This is very important. Our gene bank always, this is a contract provided by the international treaty. It is mandatory to sign this contract, so the SMTA, when the materials, I'm going to read this because this is very, very, very important. When the material is used for the purpose of utilization and conservation for research, breeding, and training for food or agriculture, you have to sign this contract. In this link, this is a very long link, you can find the text of the SMTA that we think that we will see, but you have the SMTA application. You have the link in this part, and that is very easy to sign this contract. When you have to register in this application, you have you need a personal identification number, and it's very easy to sign this contract. This is a contract between the provider and the receptor, and avoids the misuses of the plant genetic resources. Another important thing, very important thing, is when the material will be used for chemical or pharmaceutical purposes or non-feed or food industrial purposes, you have to talk with the manager of the collection or the authority or of your region. In our case is the authorities of our government, but I have to say that it's different in each country. This is very important because the access to plant genetic resources is different in each country. You have to look for the regulation of each country. I know this is a bit difficult, a bit tough, but this is the thing, are the things now. So now the next slide, the next slide is about, this is a very important thing. So you can see how difficult is the exchange of and the delivery of seeds. Another important thing is you have to know that exists a European regulation about the movement of seeds within the European Union. The most important regulation is this regulation that I have written for you. And this in this document you can find the plants and the parts of plants that need the plant passport for the movement of seeds within the European Union. You can see in this slide that the materials requiring plant passport is all the plants intended for planting, and for seeds, you can see there are some list, a list of, for example, I don't know if for you is more important the vegetable plant seeds. You can see all the crops that you need. You need the plant passport for the movement of seed. That is allium cepa, allium porro, capsicum, onion, all the Phaseolus, and also the more important crops we need. The most common for the movement of seed is Solanum lycopersicum. Solanum lycopersicum is now very difficult movement because of, I don't know if you know, the tomato brown virus. Is the tomato brown virus? Yes. So you need always the plant passport. How you get the plant passport? You have to make contact with the plant health system. I don't know if of your government, of your region. In our case, is our region. If we need this passport, also for the movement inside Spain, and of course for the rest of the Europe. And the plant passport is the guarantee that these crops are free for a list of virus, bacteria, and viruses. And for me, I recommend you, I highly recommend you, of course, this regulation that is important you have to know. And now the next one, I don't know the next one. What is? I'm going to explore. We are going to explore the. So at this moment, Adrian is tell me that we are going to explore the. See, we are going to explore a web page.
Now Eva will show you, as I told you, a virtual guided visit. Just let me a moment. Change. So, okay. Now we are sharing with you.
We are sharing with, yes.
Can you see? Can you see it?
Can you see the page of the CGN?
Yes, yes, we can see.
Yes. Okay, perfect.
Then
So I'm going to explain how to access to plant genetic resources. In this case, I'm going to explain in the case of the CGN Wageningen, because yes.
Please take notes or pay attention, because the homework will be based in this experience. So
yes.
now Eva will provide you a virtual guided visit to a repository, the one of the CGM, but the homework will be for you to visit and to work about the a repository of acid bank. Okay, so pay attention.
Yes, but I can assure that it's very easy. We have choose this the gene bank of the CGN because it's in English. We have another web page where you can ask for plant genetic resources, but the thing is, it was in Spanish, and I think it's better for you in the CGN. So for you, for ask for the material you want, you have to go. You see in this part that is search and request seeds. You enter in this part. In this bank, you have to choose the crop you are interested. For example, I'm trying for pepper. That is the aim of study of Adrian. So we search for pepper and you put pepper. Sorry, because this is loading. So the first, I would like to see when this way to access is very easy because you only have to select the crop, and we try to select. Oh, we have a lot of problem with no. We have to select pepper. Have to select pepper. It's a bit slow. So in this page, we are in this page now, and you can see the number of accession in the TGN is about 11,000. And another important thing I would like to remark is the same thing that I told you in the previous slide. Seeds supplied by CGN meet all the phytosanitary requirements of the European Union. So now we can you can go to the search taxonomic data and you can see the genus, but you can choose for a species. In this case, I am going to select Anu. You can see that I have selected Anu, and also I can choose a subcrop type. And I'm going to select Anum complex, Anus cultivated. And the thing is, if you want to plant genetic material of one specific country, for example, I don't know if you are from Bulgaria, so you can go to this and you can select this. When you have select this country, you put in this search, and you can see the number of results is 18 accession. In this part, you can enter. You can in this page, you can see the taxonomy group, the accession name, and the origin country that is the ones we have selected, and if they have a picture. The thing is, for example, if you are interested in a pepper of Kalashnikov, you enter in this part. You can see all the data of this accession. You can see the origin of the accession with the latitude and the longitude, the donor institute collecting data, and also in observation, you can see the plant characteristics of the morphological characteristics of this type of pepper. You can see that you have a lot of information, and also at the last of the page you can see a photo of these accessions. So when you have look at all this information, it's so easy that add to shopping cart. You go to the add to the shopping cart, and you can see your cart that is full. You shopping cart and go to this. Yeah, in this part you can check out and look for this. When you check out, when you check out, this is very important. This is the thing that I have said previously. You have to check if the material will be used for the purpose of the utilization and conservation for research, breeding, and training for food agriculture, or for another use. If you mark this, you only have to fill in with your information, and at the last time request these items. And also the CGN send you the SNTA for the signature. And that's all. It's very easy. This application of the CGN is very easy. I don't click in this, but it's very easy to check. And now I think this part is finished. If you have a question, we can solve it later, because I think Adrian is continue with another activity, no? So thank you. I'm going to pass through Adrian.
Okay, thank you, Eva, for such a good condensed and resilient presentation under these circumstances. Okay, I will be back to the presentation again. Close this. And please. Okay. Now, once Eva has finished, we are almost finishing. Okay. Now it's time for the fast quiz. As I told you, please answer, use your emails and send to me at doctor.ubb.es and also to Petra, both in copy. In the body of the email, the answer to these 4 questions. They are quite easy questions. So I hope you to be filled in 10 minutes. They can be answered in 5, but please do it in 10 minutes and send to both emails. And after 10 minutes, I will be back to finish the presentation, okay, of today. We will make a fast debate and final conclusions and the homework. Okay, now it's time to finishing. I will be faster because due to the CGN incident, we lost a few minutes. Okay, I will be as fast as possible to finish. Well, these are the last slides and the final part of the presentation of Unit 1. Okay, now the first point of the debate is revise the quiz in common. Okay, hope question 1. Explain the correlation between temperature, relative humidity, and cell longevity. So, is somebody wants to answer, to provide their answer? Don't be shy. Please, hands up. Which is the first hand up? Petra? Can you manage the hands up and select the first one? Because I cannot see.
Just a second. So the first one to answer your question.
Yeah. Who is the first one?
Mm.
To answer without being shy and answer the first question of the quiz?
Noemi Peter.
Okay. Let's go to Noemi.
Hello, can you hear me?
Yes, we can.
Okay, it's according to this Harrington rule that the longevity of seeds decrease by 50% if the room humidity decreases 1% and the temperature increases 6 degrees.
Okay, perfect. Thank you. This is the Harrington rule. Okay, the Harrington rule, but as an extension of the Harrington rule, you just need to remember that the lower the temperature and or the lower the relative humidity, the higher the longevity of your seeds. Okay, this is the core part. Okay, perfect. Okay,
we have
so
the
let's
same answer from Dario Danuevich, just so we. Okay.
but for the first or
Yes, for the first one. He wrote it in the comments in the chat, so I'm just reading it.
Perfect, because I prefer to pass to the second questions to be a bit faster. Okay, but yes, please put in the in the chat. Okay, the second question, maybe if Dario is wants to explain us his answer to question 2, differences between orthodox and recalcitrant species.
Maybe Maria Palardo.
Vale, Maria.
Maria.
Go ahead. The floor is yours. Maria.
Question 2.
Yes, question 2, Maria.
Recalcitrant species seeds do not support dehydration and therefore they cannot be conserved at low temperatures. But orthodox species are preserved as dried seeds and they can be refrigerated, supporting lower temperatures.
Okay, good. It's a good answer. And also you want to add, if you want to add, you can give also examples of about which kind of species we are talking about. For instance, the most of vegetables correspond to the orthodox, while most tropical species with fleshy seeds, difficult to dehydrate, etc., like for instance avocados, correspond to recalcitrant species. But your answer is good. It's perfect. Okay. Now question 3. Somebody wants to answer the question 3. Which kind levels of data are managed with PGRs?
If you want to answer the question, just raise your hand or.
Yes, the first hand up.
Maybe Fernando?
Okay, Fernando, go ahead, Fernando.
Or Sebastian, he raised his hand. Sebastian, would you like to answer the question?
Oh, yeah, sorry. Can you hear me?
Yes.
Yeah, okay. So there are 3 kinds of data. First of all, the passport data, then the characterization data, and the evaluation data. And the most common are the passport data, and then less available are then characterization data and evaluation data.
Okay, perfect. You could add also which kind of data are the passport data are the relative to the origin of the materials of the sources, while characterization data are based mainly in the international descriptors, morphological, highly inherited traits. The paradigmatic example are the descriptors, the catalogs of descriptors from the Biodiversity International, the answer EPGRI, okay? And the evaluation data corresponds to traits more specific, screenings against pathologies, abiotic stresses, or quality traits, for instance. Okay, but your answer is good. Okay, and just to finish, define in situ versus ex situ conservation. I have seen that I have a friend here for many years, Roberto Ruiz de Arcaute. So it is, don't feel coactionated, Roberto, but did you want to answer question 4?
Okay, thank you, Adrian. In situ conservation is conservation in place, in the wilderness, in the forest, the specific areas where they are original, the species. Ex situ conservation in botanical gardens and other places outside its origin.
Okay.
That is right?
Yeah, it's right.
Thank you very much.
You're very welcome. And as an example is the seedbanks, the explanation we are given today. Okay, perfect. Good. So Just to finish, I have revised the quiz in common. Unfortunately, we are on the delay because of the problems with the CGN collapsing moment. Okay, but maybe you are familiar with some seed banks close to you. I will give these questions of the debate also, if you don't mind, as part of the homework. Okay, just let me know if you have had which experience or previous experience you have, if any, with seed banks, or what do you knew before about seed banks close to you, or you know some of them. Okay, as part of the homework. And for me, it's okay for my presentation. And before going to the core homework, just leaving you the opportunity of putting other questions and doubts in the chat. Okay, so before the next session, which will start at now is 50, almost 15 past 11. Maybe we can start. Yeah, we need to start at half past, but maybe at 35. We will actually start at the 35 past 11. Okay, but to this moment, if you want, you can write on the chat also any additional questions or doubts you have during the presentation. Okay, that will be interesting for all of us, and we will ask in the next days to all of you. Okay, good. So as roll up to finish the presentation of today, just to have a fast summary on what we have to learn today. We have learned the main issues related to the seed banks management from the moment that the seeds are received in the seed bank, or the seed banks are shipping from the seed bank to the other seed bank or other users or actors, farmers, seed companies, etc. The main issue is relative to the sanitary and legal aspects, and also how they can help to support the conservation of the agrobiodiversity as a complement to the on-farm or the in-situ conservation. So they do not work as dynamically like in-situ conservation, because the resources preserved in a seed bank are not facing the climate change, the environmental challenges that in situ. Okay, but they act as security copies, which is also very important to ensure that ancient races or a huge heritage of agrobiodiversity is preserved, at least the beginning part. Okay, so this is the main issue you have learned today. Now the proposed homework, as I told you before the presentation of Eva, the proposed work for the next week to get the certificate, to get the answers to the quiz, is a fast searching of GearPlus by yourself in a repository. Maybe you can explore the CGN, okay, as explained by Eva. Yeah, okay. I will write in the chat also the email of Eva if you have any doubt. Okay, just to have extra info. Yeah, yes, and prepare and revise this part. You can explore also the other repositories. I'm also suggesting you the IURDC from Taiwan, the USDA AS program, GearPlus program. Okay, it's up to you. But please prepare a list of 20 accessions or one species chosen by yourself, but please from different origins. Try to prepare a selection of different origins, different varietal types. I mean, a diversity mixture. Okay? And please send this list of accessions with the main info you get from them in the repository. Okay? A Word file or a PDF file, copying and paste the info, it's okay for that. Okay? From now on, you have a week to finish. And better if you send directly on Monday, then we will have Petra and me will have grouped in the same day all the emails. So it's better to wait 1 week and then in the next Monday send us please the homework, and also as complement homework the previous questions I told you. If you have had some experience with seed banks close to you, please let us know. And mention us which seed banks were these ones. Okay. And from my side is everything. Just tell you that as additional available materials, we have listed a range of links, the operational GBAT manual, the PGR Biodiversity International Descriptors, where you can find a catalog of descriptors, web pages of plant genetic resources, seed banks and programs, the one from GGN, the one from Kew Gardens, the USDA-ARS program, the Real Jardín Botánico de Madrid, etc., and even our COMAP UPV seed bank. And also some additional presentations from a mate of us who has an issue on germ plants in our master science in plant breeding as complement. I'm sorry, but it is in Spanish, but I think it's also a good complement. So thank you to all of you.